Hello and welcome to episode 1 of Bio with Brad. Today we're going to be talking about cells. So, what are cells? Well, cells are the building blocks of life. They're the smallest organic unit. In this episode we're going to be talking about organelles, which are the items within cells, how organelles interact with each other to carry out certain functions, differences between prokaryotes and eukaryotes, and also how we view cells. So here we have two diagrams, however they are the same, both eukaryotic plant and animal cells. The only difference is the amount of detail put into the different organelles that are within them, and by the end of this episode you should see that they are exactly the same. I'm going to start off with the plasma membrane. They are found at the surface of animal cells and just inside the cell wall of plant cells. It's made of mainly lipids and proteins. I like to think of them as the doormen because they regulate what enters and exits the cell. When you look at them at a biomolecular level, you realise that they have channel proteins, receptors and many more things to them, but for now they're just doormen. Then there's the cell wall, which is only in plant cells, and it's mainly made of cellulose. And think of it as like the scaffolding for cells. It will always be rigid to allow for structural support so it doesn't collapse. Then there's the nucleus, which we all understand is the powerhouse and brain of the whole cell. And at GCSE level you are only taught to know the nucleus, however it has many important parts within itself. For example, genetic information is located in the heart of the nucleus, within the nucleolus and the chromatins. That is then encased in nuclear envelope, and the spaces between each nuclear envelope are called nuclear pores, and they're used for enzymes and things to go into the nucleus and source their genetic information that they need, because it contains the instructions to make proteins, which is how the nucleus controls the cell. Now here's some isolated organelles, for example the lysosome, which is a round organelle which has a membrane but no internal structure. It contains lots of digestive enzymes, and so they are quite useful when it comes to phagocytosis. Then there's ribosomes, which are very small, and they consist of a small subunit and a large subunit, and they're at the site of protein synthesis. Then there's mitochondrions, or mitochondria as the plural form. They have a double membrane, and the inner one is folded in to form structures which are called cristae. Inside of this is a matrix which contains enzymes involved in respiration. Which makes sense because it's the site of aerobic respiration in a cell, where ATP is produced as the cells can't actually use bigger molecules like sugars as energy. They need to break them down into something smaller. Then we have the RER and the SER. The RER is the rough endoplasmic reticulum. It has lots of little ribosomes along its surface, and it's involved in folding and processing proteins. The SER is the smooth endoplasmic reticulum. No ribosomes, but it's very similar in the fact that it's a system of membranes enclosing a fluid-filled space. The SER is involved in synthesizing and processing lipids. The Golgi apparatus is a group of fluid-filled, membrane-bound, flattened sacs. Its function is to process and package new lipids and proteins. This is also the site of which lysosomes are made. Then we have a plant cell exclusive organelle. It's the chloroplast. As you may know, it is the site where photosynthesis takes place. It consists of two membranes, stoma, and a granum and lamella, which are attached by thylakoid membranes. The next three organelles are made of microtubules. For example, we have the centrioles, which are involved in the separation of chromosomes. We have cilia, which are hair-like structures which push out from the edge, but the only thing that's pushing them is the microtubules within them, which are in 9+2 formation. Then there's the flagellum. Flagella on eukaryotic cells are like cilia, but longer. They stick out from the surface of the cell and are surrounded by a plasma membrane. Once again, the microtubules are in the 9+2 formation. When the microtubules contract, the flagellum moves. It's used as like an outboard motor to propel the cell, for example in sperm cells. Many organelles are involved in protein production. Proteins are made on the ribosomes, which are located on the rough endoplasmic reticulum on the outside of the nucleus. The ribosomes on the rough endoplasmic reticulum are excreted and attached to the cell membrane. However, the ribosomes in the cytoplasm make proteins that stay in the cytoplasm. The new proteins are then folded and processed, and then transported to the Golgi apparatus in vesicles. At the Golgi apparatus, the proteins undergo further processing, and then enter more vesicles, which are transported around the cell or out of the cell, and they exit via exocytosis. Organelles are surrounded by cytoplasm, but the cytoplasm is much more than just a solution of chemicals. It has a network of protein threads running through it, and these threads are called the cytoskeleton. In eukaryotic cells, the protein threads are arranged as microfilaments, so that lots of small molecules can be transported along them. The cytoskeleton has 4 main functions: structural support to keep the cells organized in position, as well as maintaining and strengthening the shape of the cell itself. These microfilaments are also involved in the movement of chromosomes during cell division as they separate, as well as being able to move materials along actively, such as with the cilia and flagellum as they waft along different materials. Prokaryotes are completely different to eukaryotes as they are a completely different type of cell. You can remember this quite easily by thinking about pro in prokaryotes rhyming with no, as in pro have no nucleus. There's many differences between prokaryotes and eukaryotes. Well, prokaryotes are usually a lot smaller, less than 2 micrometers in diameter, whereas eukaryotes can have larger cells ranging from 10 to 100 micrometers. Prokaryotic DNA is circular, whereas eukaryotic DNA is linear. Their membranes are also made from different substances. The cell wall of a prokaryote is made of polysaccharides, but not cellulose or chitin. Eukaryotes have no cell wall, as in animals, cellulose cell wall in plants, or chitin cell wall, which is found in fungi. Prokaryotes have much fewer organelles than eukaryotes, as well as their flagella being arranged in a helix structure instead of the 9+2 formation. And finally, prokaryotes have much smaller ribosomes, whereas eukaryotes are larger. It's also very difficult to look at the internal structure of prokaryotes because they are roughly 1/10 of the size of eukaryotic cells, and so you need a really strong microscope to see it. Scientists use 3 different types of microscopes to look at cells: light microscopes, laser scanning confocal microscopes, and electron microscopes. 2 important parts of this subtopic are magnification and resolution. Resolution is how detailed the image is, and how well a microscope can distinguish between 2 points in an image that are close together. However, magnification is how much bigger an image is than the specimen is in reality. We can calculate magnification by dividing the object size by the image size. Light microscopes use light. They have the lowest resolution out of the 3, which is 0.2 micrometers, and their maximum useful magnification is 1500. LSC microscopes use laser beams. In order to bring out an image, the specimens need to be tagged with a fluorescent dye. The images created are very clear because of the pinhole focus. The laser causes the dye to fluorescence, which means give off light, and the light is then focused through a pinhole on a detector and then hooked up to a PC, so a 3D image can be rendered. These microscopes are very useful when looking at objects at different depths, and especially in thick specimens. Then there's the electron microscopes. There's 2 main types, TEM and SEM. TEM, which are transmission electron microscopes, use electromagnets to focus a beam of electrons, which is transmitted through the specimen and then reflected back to provide high resolution images. However, they can only be used to look at thin specimens, if not the electrons won't go through. Then there's SEM, scanning electron microscopes. These knock off electrons from the specimen, which are then gathered in a cathode ray tube to form an image. The image produced show the surface of the specimen and can be in 3D, but they give a much lower resolution. The maximum resolution of a TEM microscope is 0.0002 micrometers, and the maximum resolution of a SEM microscope is 0.002 micrometers. And so the TEM has a much higher resolution. This is the same with its magnification. It can be more than 1,000,000 times the size of the specimen. However, the SEM microscope is usually less than 500,000 times the specimen. That's all for cells at the moment. I hope you enjoyed this episode, and any feedback you have, leave in the comments below.